Welcome to Let's Talk Micro.
Hello, dear listeners. Welcome to another episode of Let's Talk Micro. As always, I hope your week has been going great. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Amazon Music, Goodpods, wherever you listen to podcasts, you can find Let's Talk Micro. As for our social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. So either via social media or via email, you can send any feedback, any topic suggestions. They are always welcome and appreciated. So please go ahead and subscribe to the podcast, download episodes, and if the app allows you to do so, please go ahead and leave a review. Thank you so much for the support. And of course, if you're looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. Great resources from Dr. Timothy Gauthier, a very passionate pharmacist. So if you are listening to this episode by now, Microbe 2024 has passed, and I tell you, it was such a great event. So great presentations, great posters, I had a great time connecting with people. And stay tuned, there's a little episode that I recorded while I was there, while I actually talk about what I like, what I did, posters and things like that. So stay tuned. If you haven't checked out the previous episode, please go ahead and do so. It was a great episode with Dr. Madeline Barron, and she's a communication specialist at the American Society for Microbiology. She was a guest of the podcast on a previous episode about microbes in your cleaning appliances and tools. So if you haven't checked out that episode, please go ahead and do so. But in the episode, Madeline talks about what causes UTIs, what organisms are typically seen. She talks about some treatment options. She talks about what is a recurring UTI, which is short for urinary tract infection. So this was a great episode with some great information. So I'm definitely invite you to check it out if you haven't already. Well, today's episode I am going to be brief because it is longer than most. It is over an hour, but it is great content. So a few episodes ago, Grace Leu-Burke, she was a guest in the podcast, and she is the director of the Medical Laboratory Sciences program at the University of Alaska in Anchorage. And she talked about her program, and one of the things she talked about was the research that the MLS students, they do. So I invited her to the podcast to talk about this research in more detail, which is what this episode is about. And this type of research, this is something that I don't think most medical laboratory sciences programs are actually doing, but maybe you should consider it. She really breaks it down. She talked about how it got started. She talks about the testing they do, the samples, it includes like scat from bear, moose, and geese. The organisms that they're finding, what are they used to identify them? What kind of antimicrobial susceptibility profiles they're finding. And I think that one of the outcomes of this research is that the students are learning so much. They are performing so much testing. They are coming with such good concepts, and they are learning so much about antimicrobial resistance. So I think this is very positive and should definitely be considered for some other programs. The students learn so much, and also the value of research and getting involved in it. So I really like what Grace is doing. And one thing that she mentions in the episode is that that understanding that AMR is not something that it just happens in hospitals, but it's something that happens in the community, and there are so many factors to it. So definitely such a great episode. As you are listening to this episode, keep in mind that Microbe has passed. So we talked about Grace presenting in Microbe, and that unfortunately passed already. But she is going to be presenting at the joint annual meeting, which is known as JAM. And this is a joint meeting that happens yearly with the American Society for Clinical Laboratory Science, ASCLS, Association of Genetic Technologists, AGT, and the Society of American Federal Medical Laboratory Scientists, SAFMLS. So if you're attending JAM this year, which is going to be taking place in Pittsburgh, I definitely invite you to check out Grace's talk. And I am including the title of the talk and also a link to JAM registration on the show notes. So, great episode, great content. Let's go ahead and listen to it. Recently, here in the Let's Talk Micro podcast, we had a guest, Grace Leu-Burke. She was in and she talked about her MLS program at the University of Alaska in Anchorage. And, as she was describing the program, she touched on research that the students do, which is something that maybe those of you that have studied medical laboratory sciences, and I remember when I went through my program, we did like some sort of project, but like doing the type of research that she's talking about, we definitely did not do. So, and then we mentioned in the episode that at some other point in time, she was going to return and talk about the research in more detail. So, here she is to talk about the research. Grace, welcome again to Let's Talk Micro.
Oh, Louis, thanks again for inviting me here. I did listen to our first podcast together. It was so much fun, and it reminded me that I have to slow down. When I get excited about micro, I tend to talk faster and faster. My students have learned that if I start talking really fast and then if my hands start waving around, it's probably going to be on the test. So so I'm going to make a point to make sure I don't get too excited and talk too fast, because research is really the cornerstone of what we do in medical laboratory science. When we look at the lab, if you have a visitor, even when anyone comes down there, they see a lot of instrumentation, a lot of people in white lab coats, and they don't necessarily see the critical thinking that's going on. And I remind my students that even when they're running a chem profile and it goes through and everything checks out, you're really critically thinking. You're just doing it so almost automatically you're not. I mean, your QC worked, your validations worked, everything. You didn't have a delta fail. There wasn't an error code, and all of those things that you're looking for before you have even that auto release. I mean, you're monitoring that. That's all critical thinking. But because it looks so automated, I really wanted to bring that critical thinking in the education piece so that when they got out into the workplace, they would have that sense of embedded in them, because they've done it in an area that maybe we can look at a research piece. We can break down what we actually do in the clinical lab in a more bench type situation. Of course, microbiology is the most common way we can do that, because even with the instrumentation molecular that's coming in, we are still very manual. There's still things that are there. There's always new bugs. And so there's a real good opportunity for research. So when I was hired by the University of Alaska Anchorage, one of the things they really wanted to do was to bring more research into the program. Prior to my hiring in 2016, you're right, they did things like projects. They actually had a departmental honors, and we still do program where one or two students may do a project with their clinical sites, and that was always there. They've always done a capstone project, but they really wanted something different, something to really increase those critical thinking. And I always smile that it didn't come with any money. So it's not like they said, here's this bucket of money, we want to do research. They said, Grace, you've been doing outcome-based research the whole time on your bench. So it's because when I interviewed, I talked about, oh my gosh, why am I doing all this testing? Should we do something better? Or there was one weekend that I got really tired of having to recalibrate the lab-directed testing on all of the drug of abuse that we were doing serum. And I noticed that every single time we'd run a serum and a urine, the urines were always positive. In other words, the serums, yeah, but the urines always were positive. I'm like, why am I doing this? So I did a research study on this because I got tired of doing those on Saturday afternoons. And not just because I was tired, I was always curious. So when I interviewed, they says, can you bring that to the university? So it wasn't something that I already had. It's something that they wanted to see me develop, and notice I sort of had this curiosity all throughout my education. So I guess they must have trusted me that I was going to be able to develop something. And I had some ideas. I had some ideas already when I was interviewed. So we came to UAA, I came to Alaska, and I was teaching clinical microbiology and molecular diagnostics. Those are my courses. And I'm walking around, and I might have touched on this earlier, I can't remember, but I still remember passing this huge 14-inch circle of moose scat. I mean, it's just covering half of the sidewalk. and everyone's just walking around. And moose scat is like little, like pellets. They're like a very small egg, but they're a pellet size, but they're not like rabbit pellets. They're bigger than that, but they're very distinct. They're very hard. They're not soft and mushy like you think dog scat, but they're very distinct. So people think that they're fine. I mean, you technically can pick them up and they're not slimy. Not that I'd recommend that, but the consistency seems to be rather innocuous, right? So people would walk around them and nobody seemed to care. And I kept thinking, and and we're not talking like a single pile, I mean, piles and piles. And moose do not like to stand in the snow. And so they figured out that in Alaska, in Anchorage, we plow. So when the snow comes in the mountains, they migrate down. And so they're everywhere. I mean, there's technically 1,500 resident moose in the Anchorage Bowl area. And so there's these piles. And I was like, this is crazy, because I come from Kansas. We are a rural state where you move your cattle from one field to another, because that happens. And now granted, you're not going to pick up every pile, but you have to kind of clean after your cattle. That's considered a health hazard if you don't. I mean, you can't just leave stuff anywhere you want willy-nilly. And so it's sort of like, I couldn't believe everyone thought it was okay. So in the meantime, I was trying to think, what could I do that was sustainable in the research? So we don't have a lot of funding for research. I'm a bipartite, which means I don't have a research component on my contract. So I'm teaching and doing service. So what can I do to have this dream that they have? Well, we get donated media from the hospitals. And because Alaska does not have just-in-time inventory, because obviously it's hard to get things up, we get quite a bit of outdated media. And I noticed that a lot of the outdated media, which makes sense, is all for enteric pathogens, because, we're all collecting that McConkey with sorbitol, McConkey, HE, XLD, plus XLD and HE are not hard agar to make, because we have we have the ability to make Petri plates. So this is not a hard plate to make. So I'm thinking, and I'm walking around all this moose scat, and I'm like, let's see maybe what grows. And so I had a handful of students. Winter break was coming, and I could see it because it had snowed, and you can see them in the piles. And then they're cold. And I'm like, well, this is kind of good, because I really didn't, want to make sure my students are safe. And I thought, well, this will be okay. It's cold. It's on the snow. We'll put it in Ziploc bags, and we'll see what happens. And so that first winter break, we kind of played with it, for lack of a better words, because myself and 3 other students, and they collected moose. And we didn't have any, we're kind of like throwing against the wall, see if it sticks. We really didn't have any idea what we were going to do with this. And then at the meantime, Nome, one of the hospitals up in Nome, Alaska, had gotten some microscan plates, and they were not able to use them for some reason. And I can't remember if they were doing a lot number change or what the deal was. Well, again, shipping is so expensive, microscan didn't want them back, because it's cost prohibitive for them. And what are they going to do with these hundreds of plates, hundreds of microscan plates? So they call UAA, and we paid for the shipping from Nome to us. And so it's like Christmas. I couldn't believe it. I had all these microscan plates, and that's manual, right? And we could, and I had inoculators, I had everything ready, because I had a compact too, but it hadn't been used a lot, and I wasn't sure about, I didn't have a lot of cartridges at that time, but I had these. And so that's what we started out with. And we put it in some thio. We, set up like a stool culture, put some stuff at room temperature, and then I try really hard to make sure that I'm using very clean language, but I could not believe what was growing. As a clinical microbiologist, I'm like, oh my gosh, I did not expect to see a potential ESBL Klebsiella coming from a moose sample that was just laying on the ground. And you're Alaska, you're thinking, why would we have an ESBL producing? And in fact, when I saw, and of course, the microscan panels that we had were gram negatives for urine. So, they're not going to have the breakpoints but I knew from the susceptibility patterns on the cephalosporins, it was really suspicious. So I got a couple of, the discs, cephalosporin, the cephalosporin, the clavulanic acid, the classic, disc diffusion method that we all learned. You have to remember, ESBLs came on board when I was working. We did not recognize ESBLs until I told students, we had in the clinical lab, it looked like it worked. The cephalosporins looked like they were sensitive and they should work. But then we had treatment failure. And one of my students says, well, what do you mean by treatment failure? And I go, well, unfortunately, people died. In other words, we were giving what we thought were sensitive. This is the 80s and 90s. We did not know. Everything was just diffusion. We were doing some MICs, and we just didn't know what happened. And what we noticed is that when you had the change in zone between the cephalosporin and the cephalosporin with the beta-lactam inhibitor, so that classic difference in size, the reason that came about is we noticed that in our disk diffusion. We didn't do some great experiment to figure that out. It's just that we said, well, when the disk diffusion looks this way, then we have failure, clinical failure. It's almost a retrospective. And so that's why when we would do that, then we said, no, none of the beta lactams, because we just didn't know what was causing it, right? So having that background, I just did the same thing. I'm like, well, I think this looks like an ESBL because of cephalosporin sensitivity. So let's go ahead and do it. And then sure enough, I got that, the zones, and the students, their eyes got all big and they go, what are we going to do? And I go, well, first of all, you're not playing with this yet, because I was really worried because I needed to tighten up our protocols, tighten up how we're going to, because prior to that, I just figured we'd get some, E. coli and, and maybe an Enterbacter, and had no way that I thought a Klebsiella would come from a stool sample. Now, as we all know, fecal matter comes from what you eat, and moose eat sticks and, branches and plants, and they don't eat meat. They're not a carnivore. So I wasn't surprised we didn't see E. coli, but I didn't know what we would see. So that's how it started. And so I sat the students down and said, you know what? We're going to need to make a team, and we're going to need to do more of these. So instead of us working way too hard after class, we're just going to have advanced micro do it, because the spring semester is advanced micro. So I rapidly switched all my enteric pathogen fecal cultures that I normally were mocking up, and we just did these stool cultures. So we sat there and thought, well, let's just collect everything cold. We'll keep it cold because it's cold, and then we'll thaw it out during that session and we'll see what grows, because we started thinking about what would that affect the patient ourselves. I mean, how is that going to interact? And so we started doing a lot of reading on environmental exposure, a lot of reading on transmission of bacteria from animals to humans. That's not something we really focus on in clinical micro. We always assume the bug is there in the human, or, close proximity, but we don't really think about zoonosis in that way, especially bacteria and colonization. And so the 3 students and I worked really hard just trying to hurry up and figure out what to do, because we never expected that within a first week of culturing, we'd have these significant antimicrobial resistance. And I told our students, I said, not only is this really important, interesting, it's really important, because I don't think anyone expects this. People are making jewelry out of this stuff. This moose scat is something that's sort of a tourist thing. And, we right away think this is not a tourist thing. And so we already started talking public health, and that's how it started. And we sat there that spring semester and figured out a protocol, because I went online, I did a literature review, and nobody is culturing moose scat. There isn't a protocol set up. So I told our students, we need to establish this now so that we can compare, and we're going to do a surveillance. And so all of a sudden, this is going to be the research that UAA MLS students are going to do. This is what we're going to be known for. We're going to analyze this moose scat and see what happens. And so it started from there, but I didn't really expect that it would be such a significant finding. I really didn't. And mainly because it's organic, right? We're not feeding moose antibiotic-laced feed, right? They're just hanging around because they don't want to stand in the snow. So that's how it started.
Wow, that's fascinating. And the thing is, so you said that they normally, so people do make jewelry Like they handle it like that?
Yeah, they will like shellac it and make it into earrings and stuff. So moving fast forward, we've got a lot of data. So at this point, I don't have an exact number, that first 2017, 2018, some of that data didn't really know what we were doing. Not that we were not being careful, but that consistency that you need in research. And you have to recognize we were building our own protocols on our own. But I had presented at the One Health conference in Fairbanks, and that would have been 2022, 23. Okay, sorry, I can't remember what year it is. So 2023 we did, and it was great. It was an international conference, and we actually spoke. And at that conference, the Alaska Beacon had, well, there was reporters, and it was a very large conference, and it was international and online, so it was really great. It was really great. It happened to be in Fairbanks because we're in the middle of circumpolar. So I told my students, they go like, well, how can it be exciting conference? It's in Fairbanks. I'm like, well, because guys, we're circumpolar, we're the Arctic. That's why it's in Fairbanks. So it was lucky for us because we could travel to it. And it was a phenomenal conference, and we were picked up by the Alaska Beacon, and then we were interviewed, and then the news people interviewed us, the local Alaska, and that's where I finally got to say, please don't use it to make jewelry. And it was interesting to take the data that we have as scientists and talk about ESBL and carbentate paste producing and all those words we just rattle off, right? And to make it so that the public is not scared to go outside, because our data points that in the Anchorage Bowl area, which is about 30 square miles, is that 71% of any moose scat that you come across has one or more multi-drug resistant organisms in it. 71%, no matter where in the Anchorage Bowl that you're walking or hiking. So I did not want people to be scared to go outside, because it's still, I mean, you still have to have somewhat close proximity to be able to colonize yourself with bacteria. I think I'm more concerned about dogs because I'm always worried about when animals dig and they produce aerosols, and they don't realize that you don't really breathe in that, but you'll swallow it. People don't realize that, when you're digging and that you actually have particles that you do swallow. And that is one of the more common ways to become colonized. And so I do worry a little bit about our domesticated animals, and they're digging. Of course, they're always going after that moose scat pile because I'm sure it smells wonderful to them. So that does concern me to some extent, but I still wanted them just to be aware. And one of the big things was don't make jewelry out of it. But the one thing we also discussed as a public health is that individuals were using it for composting because you think it's natural. And actually, because they eat a lot of sticks and stuff, when you break apart that nugget, it's almost like this granular, grainy. I mean, it's not soft. Again, it's very dry. And so it almost looks like something that would compost, or they would mow over it. And I would say, don't do that, because when you do that, you're producing, first, if you mow over it, you're going to produce an aerosol, right? Because dust and that, and you could become colonized. And if you compost it, our concern, and that's what we're moving towards in this next couple of years, is that it will become part of the soil. Because not only have we picked up, not in moose, but we diverged to bear scat, geese scat. So we do more than moose scat. We started with moose scat because that was the first thing that I noticed, but we have diverged, and our bear scat. And during the pandemic, bears became more in our populated areas because, we were no longer there, on the streets as much. They became more comfortable in the areas around humans, and we still they get in our garbage. We know we're very careful. Alaskans are very careful. We really know to be careful, but let's face it, bears are bears. And because of that, we have been concerned about E. coli 0157, Shiga toxin producing E. coli. And of course, we all know as microbiologists, and bear scat's more like the traditional scat that you think of, soft, mushy, and so it'll dissolve in the soil. And we have picked up 0157 from bear and scat that we found. And of course, we're concerned that once you use that for composting, it becomes part of your soil too. And so when it becomes part of your soil, there's always a potential it could become part of your plant. And so that's another transmission. So one of the things with our moose scat, like I said, don't make jewelry. And they actually said, don't compost it. One of the things we recommend is that you just, rake it up, bag it, and toss it away. Don't treat it like a organic, natural, very benign thing, because it really isn't. It really isn't.
So I wanna talk a little bit about your procedure. So now you say that after the initial research that you were doing, did a procedure, and we talk about media. So we're thinking about stool samples, and we think about media, right? Like Hecto and XLD. So can you talk more about the procedure? So you get the samples, you plate them, things like that.
Sure. So our procedures, one of the things we wanted to be certain was that we were consistent. It all starts with specimen collection, right? Specimen collection is the key. So we wanted to make sure that we were doing things consistently, specimen collection. And then not only for environmental cultures, because we are environmental cultures, so we do not have IACUC, we're not integrating with the animals. This is not an animal project, it's an environmental project. So we have real strict guidelines in collecting. They're ground collected, they're not collected directly from the animal. And then where they're at, in the very beginning, we were doing GPS coordinates because my students really like that. And then we realized that that was really not important. What was more important for us was human and animal and soil interaction. So in other words, what pocket areas where we would have the highest impact, where a human could come into contact with these piles. And so we also wanted to be able to do what's called citizen scientists. We wanted to have anybody collect if they wanted to. So that's why we moved away from some of the Carrie Blair collection kits and actually went with Ziploc bags. And when it came for stool samples for the moose, we want 4 nuggets, very specific 4 nuggets. Anything, if it's a geese sample, get as much as you can because those geese samples are pretty small. And then for bear, about 2 tablespoons. And, we would take size because I once had, when they were collecting, I had a really conscientious faculty member that within our allied health, she wasn't med lab scientist, but she was in one of the other allied programs, and she wanted to make sure she got enough sample, and she gave me this huge bag of bear scat. So I don't really need that much. So very specific with how much. Ziploc bag and keep it cool and out of direct sunlight. That's all we ask for. It's Alaska, so not everybody's mom's gonna let it put them in the refrigerator. So cool garage, where no direct sunlight and not where it's gonna get warm. And then try to bring it to the lab within 24 hours, and that's our goal. And so in the lab, we have a bucket that's in the refrigerator, and so they're placed in the refrigerator for our collection. We collect moose scat in the spring, and we collect moose scat in the fall. So we have 2 times of the year we collect. Part of that is for 015 shedding, because 015 sheds sometimes in the spring and sometimes in the fall. So we're always looking for 015-7 in moose, not because we think they're going to start eating, garbage, but because once we pick up 015-7 in the moose, then we know it's in the plant, right? Right. That's our direct. And when it comes to bear, same thing. We collect bear in the summer because they're hibernating in the wintertime. I always tell my students, be careful, because if there's bear scat, there's bear. And so really be careful about that. So bear scat, again, it kept cold. When I come back and check in August, what's all been collected, not only by me, but students and community, then we freeze them. So why we freeze them is that we want to make it like the Alaska winter. So the scat's on the ground, it gets frozen. So this is not an immediate interaction with it, but what happens when it's frozen and then the following spring, our moose and bear scat's frozen, we will thaw it, and then we'll put some in Carrie Blair for our enteric pathogens, plus it helps us to maintain it when it's at. And then we just do a thiol broth 24 hours. We put about a pea size. It's hard to kind of measure. We just sort of, I actually do this part because I don't want the students to be dealing with potential aerosols. So I will inoculate all the thiol broth, and then it incubates for 12 hours. And then they subculture it to all the plates, including the enteric pathogens also. So we do enteric pathogens not only in the when the initial sample in Carrie Blair, but also a subculture, because we kind of want to see what's there. Typically enteric pathogens happens when you field dress the moose or field dress the bear. And so that's where the highest concentration of ability to get sick from them, or the meat gets contaminated. So we recognize that we're not getting true enteric pathogen data, because ideally we'd have rectal swabs or entrail swabs. So we get that. So we're just sort of looking to see what's there. What we think is important is that if there are potential enteric pathogens in the bowel, if someone would handle the fecal matter afterwards or be around it, again, the risk for colonization and that. So we recognize that some limitations with that. It's not a true, what we would consider research for enteric pathogens as a immediate slaughter position like you would in a slaughterhouse or a meat processing plant. So we recognize, because again, we don't want to be dealing with animals. We are dealing with environmental cultures, but we get some data that way. And then we set up, right, McConkey, agar, XLD, HE. We will do a Shiga toxin induction broth. We do McConkey with sorbitol. Then we also do room temperature for Yersinia, so CIN or Yersinia agar, whatever we have. We also do a McConkey agar at room temperature. One of the reasons we do this is that we're coming up with some really interesting different gram negatives, and this has sort of evolved because, I tell my students I'm always looking for the potential for emerging pathogens. And there's always that where that bacteria is growing at room temperature or is available there in that soil sample, that water sample where close human contact. And so their potential to become colonized or infected with it. So I always tell students it's not a bad idea to do room temperature. Plus it's really great because they're unusual organisms. And so I don't have to mock up an unusual organism. They get some unusual organisms. And so, and actually we're looking at one, and I don't know how to pronounce it, so I'm not even going to try. But it's a soil water organism. But later on, we've noticed that it's an actual potential pathogen that they're looking at as a contamination in the hospitals. But of course, there's no AST. There's no antimicrobial susceptibility pattern yet, because of course they haven't had enough. And we isolate it in about 20% of our samples. And so I told my students, we're going to start doing an antimicrobial susceptibility pattern. We're going to start running it and see what our susceptibility patterns are, what our MSC breakpoints are on this. And so to get that data, right, to have that. So in case it would become a potential pathogen, we'd be like, hey, UAA has been growing this. So that's what we also do. So we do that. And then of course, also Campy. And we don't do Campy thiol, we just do Campy plate and look for Campy. So we're looking for Salmonella, Shigella, Yersinia, E. coli 0157. And what we'll do is on any of the non-sorbitol fermenters, which again, because these are animal fecal samples, we get non-015, we get non-sorbitol fermenting, and they're not E. coli, there's something else. So it's a really good experience for the students to know, just because it's not sorbitol fermenting, remember, that whole tree says you got to confirm that it's E. coli, right? Well, they're like, oh, it's not E. coli, because then they'll take that non-sorbitol fermenting, they'll sub it to a split plate for McConkey and sheep blood, because I get tons of split plates from the hospital. I think they set up urines on them. So I've got a McConkey on one side, I got sheep blood on the other side. So they'll sub it to that. So we've got a nice isolated colony. And then they do the indole, and then they do the spot PYR, and they can identify E. coli just like that, which is great, because then they'll see on the regular McConkey, it'll be lactose fermenting, right? So lactose fermenting on the regular on the regular McConkey, split plate, they're doing the indole, they're doing the PYR on the split plate, and then of course non-sorbitol fermenting on the McConkey with sorbitol, and they go, oh, potential 0157, and then we do the latex. Isn't that great? It's exactly what we do in the clinical lab. Very difficult to mock up. And the reason I have my advanced students do this and not my 200 level is because they need to have good techniques. They need to know how to do an indole without worrying about, they just need to make sure that we're using good laboratory techniques, and they know this. They know that they could have potential 0157. So their safety is right there. You can just tell the difference. I mean, they're always careful. My students are great. They really are good students. But when they know that they don't know what's in there, because I don't even know what's in there, that whole level of, I can't touch my face, I have to wash my hands, I have to make sure that I have everything clean on my counter, that I can't cross-contaminate is there. So that's what we'll do. And then, of course, if for the XLD or the HE, again, if you have a H2S producer and it's non-fermenting, we go ahead and do TSI slants, right? We'll do a urease slant, urease positive, we're like, no big deal. If it's urease negative, then we know that we potentially have a Salmonella and Shigella. And so at that point, then we will, sometimes we'll do an API strip, but more than likely we will just do a presumptive ID. Then we'll run the antimicrobial susceptibility testing on the VITEC, because I've got a lot of not identification, but a lot of AST for the VITEC. One of the hospitals were generous and donated a lot of them for us. So we're able to do that. Then it's really antimicrobial resistant. So in other words, I really want to have a good genus and species. I'll figure what salmonella species. I can put that in the VITEC. It'll give me my interpretation. And I'm like, whoa, we need to really identify this. Then we'll go back and we still have microscan panels that have the biochemicals, so we can do a better ID. If it's something that I get really concerned about, I freeze it. And when I'll get API strips and we'll do that. So we're very much, but most things we can identify, because most things are, Klebsiella, and we do motility, and it's all the basic biochemicals. So the students will do all of that. So besides the enteric pathogens, we identify any E. coli. If it's an 0157 or not an 0157, we will still do an antimicrobial susceptibility test. We'll do a disk diffusion first, because I want the students to do disk diffusion practice. We'll do a disk diffusion first, look for that potential ESBL, and then those that are highly resistant, we will run on and verify on the VITEC. And so we'll confirm with that, but they're really good at looking at those disk diffusions and say, oh, I have a potential ESBL. If they do, then they do the disk diffusion confirmation for ESBL. And so they do that. And so that makes it really good for the students, because they are not just looking at the antimicrobial susceptibility testing results. They're already looking at those key cephalosporins, those key, when the zones are less than this, that potential ESBL. It's already in the back of their head. So although I tell them the computer will tell them, they need to know. They need to be able to scan at those results and know what they have. So all the E. coli's then are identified, all of them. So they have learned to look at a mixed culture plate and pull out the lactose fermenter subculture, again, indol positive, PYR negative. Sometimes we get an indol negative, PYR negative, realizing that there is about 5% of the E. coli's are indol negative. So they get that too. So we always verify that. We'll do a citrate and urease. Why not? Citrate and urease slants are easy to do. E. coli is very non-reactive, right? They're like Shigella. So it's really great. It really enforces some of those just spot biochemicals that you can verify, so they're not having to rely on a huge API strip or something. So we'll do that. And so when you think about it, it takes 2 weeks to go through those cultures. Now, for gram positive, so we've only done gram negatives, and we started with gram negatives, and we did gram negatives for a couple of years, but I really wanted to do gram positives. I had a couple of students that were doing departmental honors, so they helped us to create that protocol. So what we do then, same thiol broth, but then we sub it to Columbia agar. We sub it to any chrome agar that we have. So we have chrome agar for like MRSA and VRE, so we'll put it to there. And then we will also do a sheep blood agar. And we'll do a macaque, just so that we have a feeling about how much normal flora is there with the gram negative, gram positives. It's a little bit easier when you've got Columbia for your gram positives and the macaque for gram negative. We can make macaque agar really easily. And so we start looking, and we only care about staph, which we don't grow very often, which amazes me. I can't tell you how many cultures we've done, and we don't really pick up staph. But we do pick up alpha hemolytic strep. And so we'll pick up the alpha hemolytic strep, bile, echelon, and salt, just like you do for enterococcus. Anything that's enterococcus positive, then we can do an ID and sensitivity on the VITEC, and then we will do that. And we're looking at vancomycin-resistant enterococcus fecalis and fecaum. We look at linzolid resistance, and then we also will see what else we see. So we get enterococcus scalarium, enterococcus calciflavus. It's really good for the students to see that that type of sensitivity pattern, the idea of intrinsic and acquired. And it's okay, we in the clinical lab, we're always so focused on, vancomycin-resistant enterococcus fecalis and fecaum. But, if you have a shared environment and there's an enterococcus scalarium or an enterococcus calciflavus, and an individual becomes colonized with that because they are camping in an area of housing insecure. So they're camping and shared environment. So they could be colonized with that type of an enterococcus. They could get a urinary tract infection and then subsequent sepsis, because that's one of the number one ways we get sepsis is an uncontrolled urinary tract infection, and it could be an enterococcus calciflavus. So for us to sort of think, oh, that's not a true pathogen, we don't see it, we have to really remind ourselves that because we have environmental exposure, then we have environmental opportunity for colonization, then we become a reservoir for some of these organisms that we would consider unusual, and they probably still are, but to assume that we shouldn't be looking for them or doing research on them is why we still identify them. So even though they're not truly a pathogen, as we see, they're not something we look at in the clinical lab as far as, surveillance, but it's something I think it's important. So the students, and it's great because they know their alpha hemolytic reps. I mean, how many students do you know graduating from MedLab science program can just rattle off Cassiflavus? They just know it because they isolate it. So we do that on all the isolates on both the moose, bear, and then the geese. Now the geese samples are handled a little bit differently because they're small and we keep them fresh. So I don't know about you, but the geese seem to defecate on our entire parking lot. So these are our 200 level students. So to get my 200 level students involved, they work on the geese samples. And so same thing, they collect them in Ziploc bags, but after they collect them, they bring them to the lab. We go ahead and put them in a thiol broth, and then they also direct plate. So they'll direct plate to the enteric pathogens. We don't bother with the Carrie Blair, they're such small samples, but then they also will incubate the thiol. And then the thiol, they will do enteric pathogens again, and then the gram positives. And it's really good for them because not only does it work on that direct plating, isolation, getting those individual isolated colonies, so really good learning. Geese is not nearly as dangerous. We do see some vancomycin-resistant Enterococcus faecalis and faecium, but not very often. Get some, but we don't see the ESBLs as much. So I'm not quite as concerned, but of course we're watching. And this is about halfway through the program. So they've had a good section on lab. They've gone through their gram positives, they've gone through their gram negatives. And so this is like their first mixed cultures. That's what they're doing. So they'll set them up. But again, the same thing. We don't worry about, at this point, E. coli 0157 with the induction broth because we don't get a lot. We have to purchase the induction broth. And so I kind of really do that for those organisms, those animals that we would actually eat. So the bear and the moose we actually eat. We don't eat the geese. We do the macaque with sorbitol. So we do that, but we don't do the induction broth. And on the moose and bear with the induction broth, if it is cloudy, we treat it just like you do an induction broth, where we just go ahead and do we subculture to make sure it's E. coli, and then we do an EIA method to see if it's the Shiga toxin, if it's the virotoxin 1 or 2. So we actually do a latex on the broth, just like you would on a regular hospital induction broth procedure. So we do that. Does that, I mean, it's a lot, and then of course we look at our campy plates, but I will tell you, we never grow campy, but I make the students do it. It's always a good thing. I've yet to grow campy, even on all those geese samples. And I don't know. I know we've done at least 400 moose samples, but we probably have done at least that many geese because the geese is easier. We set up a lot. We have a lot more samples, and we have never grown Campylobacter. So just letting everybody out there know, I have yet to isolate Campylobacter. Not for lack of trying.
Yeah, and I think even in the lab, I mean, nowadays it's we do mostly, like most stool cultures, that have been replaced by molecular tests. And then if you get something positive, then you will go ahead and, like, for salmonella, you will plate it, or shigella, things like that. But campy sometimes it's hard to isolate in the lab. It has been difficult. So when we do get it, typically nowadays, it's been detected on the molecular test. Um, wow, listening to all that, putting all the research aside and everything, they're getting so much knowledge and such a rich experience because, that's what micro is about. By doing all those tests in biochemicals and looking at the cultures and subbing them and looking at that morphology, that's how you build that knowledge. So they're coming out with some really strong knowledge when they go into the workspace. I mean, that alone, it's great. And
I'm amazed how much my advanced students understand antimicrobial resistance. I mean, when we were before, the first year I was here, I did the traditional where you have some samples, you can get some, organisms from the hospital, some of the ATCC ones, but this is different. And because we don't always know, and they see lots of sensitive stuff too, just like you do in the clinical lab, but their ability to talk about carbenipase, their ability to talk about ESBLs as if they know it. And for that level of micro knowledge, especially antimicrobial susceptibility, is amazing, because that's very difficult to teach. And I really admire microbiology instructors, because I was struggling it with myself the first year. But the minute that they have these cultures and it's right in front of them, maybe it's the relevance, maybe they know that this is part of research. So they know these are not mock-ups. They know that we don't know, I don't know, and they need to make certain that we're doing it correctly, because they are part. They might not, their name may never be on a paper, but they know that their data, they'll do 5 moose cultures. That's a lot of mixed cultures that they're working on. And when they're done, they know that that information is going to go to this group. So because we're generating a lot of data, I decided to create a MLS, what I call research team. And everyone's eligible to be on the team as long as they're in the MLS program. And so it allowed maybe some of our 100 levels, the students are just getting started, to start logging in, because it's a hybrid. So we are on campus and then it's Zoom linked at the same time. During the summer, it's all Zoom linked, because I spend a period of time back at our home in Kansas. We still own a house and land in Kansas. So we're reverse snowbirds for Alaska. When everyone's enjoying Alaska, we're in Kansas in the heat. It's the way we are. But we meet at least every other week during the school year. Depends on my schedule, the student's schedule. And we talk about what we do. And the students at first, when they think it's a research team, they think they're going to do more lab work. And I'm like, no, research team is about data analysis. It's about literature review. It's about how our protocol's good. And it's a roundtable discussion. I have facilitated, of course, because, I kind of want to guide the direction to some extent. But it's so much about, what do you think this means? What do you think this printout is? We'll look at all of the Vitech printouts. And this summer, they're on Teams right now. We'll use Google. They have like their Google folders, and they're working in teams and starting doing literature review on areas and trying to find out what's out there. What do they find environmentally? What is going on? How is it applying to us? Where is our data going? Where are we concerned about? That's why we've moved also to parasitology. So we actually, on our moose, bear, and geese, we do that little EIA kit that you do for Giardia and Cryptosporidium. And then we also do a modified acid fast on all our samples looking for Cryptosporidium. We're identifying a lot of Cryptosporidium that are, your acid fast, modified acid fast, that are not necessarily EIA positive. So that's a whole nother topic. I've got a student that's looking into that, is that, we assume that Cryptosporidium parvum is the one that causes infection, but is it the one that's causing infection because it's the only one that we're picking up? Isn't it possible that Cryptosporidium, that, again, we're talking about our housing insecure. I mean, this United States, our area, we have a lot of people that are living in areas where they do not have clean water, that they're living in shared environment with the environment. And how do we not know that individuals coming to the hospital with diarrhea and cramping that we're attributing to poor diet, maybe alcoholism, whatever, they're really got Cryptosporidium, right? And it's just that we're not picking it up because we're not looking for it. So that's a side project that came about a couple of years ago, that I have a couple students that were really looking at it, looking at our protocols, doing lots of modified. So my students can read a modified acid fast name without a problem. I mean, that's something to do one slide and everybody demonstrates. It's another one to be doing 20, because they got to do 10 and they got to do 2 of them, right? 2 slides per 10. And then they got to do one of their partners, so that we have QC. That's how you do QC on those, right? Somebody else reads your slides. And so we're doing that. And so that's part of that process too. But so we created this team. Anyone can be part of it. And that's the team that does more of the writing, reading, presenting to conferences. So you don't have to do that, but you have to do the research data. One of the things that I always felt significant about is access to research. And so many individuals think that, especially students, think that if you want to do research, you've got to be that straight A student, or that 3.8 GPA. And if you look at a lot of applications, sometimes 2 projects, even our departmental honors, requires a minimal GPA. And I tell my students, when I talk to them about being part of the research team, I tell them, first of all, you can drop in and drop out. It's not like once you commit to it, you have to commit the whole semester. I mean, you have lives. And then I would say, and there's that professor that's always giving you tests. You have to, continue to do well. So if you just want to know what's going on, just log in and listen or just come. It's only when you get really interested and you maybe want to help lead a project that I ask for a little bit more time commitment. But up until that point, you can just log in and log off. And so a lot of my students that haven't even started the micro will start doing that. I have a student this summer that's doing that, and they start getting interested at the very beginning. And I had one student one year that decided to do that, and he was truly a BC student. Chris is a really great kid, but some kids test better than others, right? And he did great. And he was a whiz when it came to looking at Excel files. And there was one day that he really wanted to lead the project on E. coli. And I said, Chris, you're struggling in school. You need to make sure that you're getting those grades. He says, yeah, but I want to do this. So winter break came and I had all these Excel files on E. coli. We've been collecting random data on E. coli and I hadn't done anything with it. And so I'm like, here, Chris, we have all this E. coli data. Just if you're going to do anything over winter break, do it. Well, he did and found that there was a significant trend in resistance from 2018 to 2021 on E. coli. There's this trajectory. I couldn't believe it. He emailed me, professor, said, we need to have a meeting. And so he showed me the data that he had. And I'm like, oh my gosh, Chris. And he said, yeah, so we got to get this together. And because of COVID and that, the ASCP conference allowed a delay for some of the abstracts because normally, and I said, we have got to get this. So we worked hard, the two of us, and then his sister was also a student. And so we brought his sister on board. I always like to have at least two students. And we wrote it up. And it wound up being one of the top winners at the ASCP conference that following October. And we submitted it under professional, not under undergraduate research, under professional guidelines because it was so significant. Okay, so this is a kid that's a BC student. This is a kid that's an MLT graduate. Now, when are you going to see in a regular university setting where someone that's a BC MLT that's going to submit an abstract to an ASCP conference? And what did it for him is that he could not believe that I have this value as a researcher. I go, yes, you do. I mean, research is not just lab. Research is, what does this data mean? What does it look like? You could see the pattern. You saw the pattern. I didn't. I just gave you a bunch of Excel files. And that's what I want my students to come away with. That is what I want my program to be. And when you have that type of open access, then you have that. Anytime there's an application, and we still do departmental honors, there is a place for that in our program. This doesn't replace that at all. And often my departmental honor students are ones that I really need them to do a little bit more work and lead on something because I really like the Cryptosporidium, the original Enterococcus. Those were departmental honor students. And so, yes, they have a place, but to be able to have an opportunity for somebody that just is curious and can be part of this is when we start getting diversity. And we would never have had that. I would have never had that abstract, would have never gone if this kid hadn't said, I've been listening to this. I really think E. coli is cool. Can I do this? And I said, sure. And if he had to apply for that, that wouldn't have happened. And so that's always my success story. And I've had a number of that, but this is what I think we need to be as an MLS program, because now he's in that workplace and you and I both know he's thinking, do I really need to run this test, right? Is there a better process? Can I do a better process? Can we maybe set up an LDT that's better than what we're doing now? Because he had that opportunity and was given that yes, right? We want to give our students that, yes, you can do this. And when they don't realize, they're not too scared to try, right? When you get in the workplace, it's scarier to try something new because now you have bosses, there's protocols, it's scary. But in a teaching environment, it's a little easier for me to tell that student yes, and they'll go for it because the level of error is, the consequences are lower. So that's my story.
It's a great one, and yeah, and then he's also going with those critical thinking skills, which are so important as part of what we do as medical lab scientists, and that's always great. And I think sometimes some techs are a little afraid of exercising that, but that's just, it makes you better at what you do, and you're thinking, okay, maybe what can we do better here or there, and that's always at the end, it just benefits your patients. Um, so you mentioned so as you were talking about some of your findings, you mentioned that you recover like a ESBL. You talk about also a CRE. Maybe if you can talk a little bit more about the results that you had.
So once we started, when Chris came up with that, I recognized that we needed to look at over a period of time resistance patterns. And two things happened as we were gaining more success in in our research. Because again, I'm bipartite, so I don't have time to write grants. I did get seed money from the ASCP. They had a grant for program directors. And so I was able to start the program with this. That's how we actually initiated, got some of our Shiga toxin reagents that we don't get from the hospitals. And then the university, my department, really recognized the importance. So I really give a shout out to UAA, Allied Health, College of Health, my deans, my assistant deans, recognizing the importance of this. Even our provost recognizes the importance of this. You know that it's important when the assistant dean of the College of Health is collecting, caribou scat when they're out hiking in Denali. So you really know that when your assistant dean becomes a citizen scientist, that they have really bought into this really great thing that we're doing here. So we got a little funding, which helps. One of the biggest costs, believe it or not, is, well, we would understand as microbiologists, is biohazard, removal. We generate a lot of plates, a lot of biohazard material. We have to be careful how we dispose it. So they were able to do that. What we were finding, and this is what our concerns are, is that when we also were able to get a software upgrade on our Vitek. So we had a Vitek Compact 2 that was put in, I think, 2012 and very manual and had not had a software upgrade. I was working closely with our BioMoreau, she's their technical support. She comes up and visits and she always comes to see us. She always says, I see your kids everywhere, when she does Alaska. And so we were talking and she says, you probably need a software upgrade. And of course, that doesn't come free. And BioMoreau but they had a program, they were doing a software upgrade. And so we could be part of that program. So I went to the university and we wrote a proposal for the department and they funded that. And, it was a good $10,000-$15,000 between software upgrade. And then we went ahead and did a PM that hadn't been done for a while because I really was concerned about. So once we got that software upgrade, any sample that we had that I was really concerned about that looked like an ESBL on disk diffusion and what I could see on our microscan, but I couldn't confirm it. We're not sequencing. I don't have any ability to look for any kind of gene sequence. I freeze. I'd have beads. I bought some beads and glycerol, so I freeze everything. So when we got the software upgrade over spring break, I thawed and I, resubbed cultures of ESBL, Klebsiellas, Hafnias, Echinobacters, and I ran them and I was seeing the warnings from Vitek, carbenipase, potential carbenipase, potential CTM. I mean, because it will give you that. And I went, oh my goodness. And so we went back. And so what we saw is that in the very beginning, we had an ESBL and probably carbenipase producing Klebsiella from the very beginning. We started seeing Acinetobacter, and I don't have my data in front of us, but it grew over a period of time. And now, since last year was the first year that we saw an E. coli that was ESBL, but also a carbenipase producer. And again, you have to remember, we think of E. coli as a really common bacteria, but in animals, it's not necessarily. We still only isolate maybe 25 to 30% the most E. coli out of moose, because again, the plants that they eat, they're not going to. So a lot of times what I think of is when they're colonized with the E. coli, it's more of an environmental colonization and not exactly a food colonization. And so, but of course, bear eat garbage and that. So not surprisingly, that our bears are coming up with more antimicrobial resistance. And in fact, when we do bear samples, it's the one time I actually will use the ESBL chrome agar. It's a little pricey for me, so we only reserve it for bears because we tend to get more antimicrobial resistant E. coli from bears. I don't want to miss one. And because you could have a mix of E. coli, with some of them being sensitive E. coli and other E. coli being, obviously it could be an ESBL. So we do use chrome agar for our bear samples and pick up that. But yeah, so now every classification, so we have a Citrobacter, Hafnia, Actinobacter, Klebsiella, Enterobacter, and E. coli that are all a ESBL producer and a carbaryl paste producer. So all of those 6 classifications of organisms we have isolated in at least one sample since 2018, but not until 2023 did we have where everybody got hit. So everything was positive. And when I say 23, so this spring we did samples collected in 23. I remember all our samples are a year old. Remember the initial collection we collect in the spring. So like we collected the spring of 24. They're going to be frozen over the winter months. And then in the spring of 25 is when we're going to thaw and run them. So our samples, so when I say 23 means that we identified them this coming spring, this last spring, because they were, so just remember everything's like a year behind as far as collection dates. And that's one of the, I'm going to speak at ASM and that's what I'm going to discuss. So we don't have a real good confirmation because of course we can't sequence, ideally you would sequence these. So there is a method where you can actually take a meropenem disc and you can inoculate it with your suspected carbenapase producing organism and see that if it will actually basically turn it off on the meropenem disc. In other words, it hydrolyzes the meropenem. And then we take that disc and then put it on an E. coli that should be sensitive to everything. And then if it's resistant, then obviously we hydrolyzed the meropenem drug in that disc. When you think about it, really cool cause and effect. And so that's what we're using to make sure, even though everything is kind of like looks like a duck, acts like a duck, it probably is. So we do that, and every one of them that the Vitech flagged as a potential CRE did actually do that, where we could show that the meropenem was hydrolyzed and that it was no longer able, even though it should have been nice sensitive disc on an E. coli, it didn't. It looked resistant, so there was no more antibiotic in that disc. And so we've done that. That's we actually came up with that procedure. We saw it online. I think ARUP Labs. I can't remember where we saw it. We went ahead and did it and QC'd it, made sure it worked, and validated it as best as we could. And it's good. I mean, it kind of, we always say presumptive because we can't sequence. I tell my students, this is what we see. This is what we potentially see. We can't sequence these organisms, but we can say that we have a highly suspected. And because of that, again, potential for environment. We don't say where it comes from. We don't say that everyone's going to be colonized. We're just letting everybody know it's out there. And the one thing that we talk a lot about with our students and with people that we visit with is that we have to stop thinking of antimicrobial resistance as something that is just happening in the hospitals. It is an environmental action. And I think what's neat about our program is we're actually growing it. So we're taking samples that have been frozen and then thawed. And sometimes the only thing that's growing is the antimicrobial resistant bacteria. So in other words, it's not a bunch of mixed flora and we're isolating 1 or 2 colonies. It's 3+ of this Klebsiella, and it's the only thing sliming off our plate. And that means that after winter and that thaws and it gets in a warm, moist environment, it's growing that. And that's what's concerning to me. And that's what we look at. We look at that potential. I do want to mention, because I keep forgetting, is that I wanted my students to be able to express this in a symposium, and not just things that the research team is doing, but also students that maybe could only drop in and drop out, or they want to talk about, a certain thing that they do. So we do a research symposium every January. It's the first weekend after school starts. So after Martin Luther King Day, we always start on that Tuesday. So that following Saturday, we actually have it, and it ties in with our advisory board meeting. So afterwards, then the advisory board meeting, after our advisory board meeting, they can stay on and watch the symposium. It's usually about 2 hours in length, and the students present. I make nice invitations so that they can take them home to their parents at Christmas time or winter break so they can put them on the refrigerator. I always say the moms want something on the refrigerator. They want to know what you guys are doing. And they present. And that's all student presentation. I often do an introduction, a little bit about where we're going with the program, and then the students do that. And we've had people log in from all over because, again, we're going to be known. And I will tell you, Louis, our biggest excitement is that just recently, one of our abstracts that we sent to a conference was published because, you know, when you are under professional guidelines, they are published. And we were cited in a One Health journal just this last February. I told my students, there we are, number 4. And so because they are saying, you know, there is a risk of E. coli resistance, right? And then they cite UAA because UAA has been phenotypically growing it. And they cited our work that we do. And I thought that was really exciting for my students.
Wow, nice. Congratulations. And yeah, well, that abstract, if you can, I would love to look at it if you can tell me where I can find it.
Yeah, and that's the other thing, Louis. They always say, well, where are your papers? You know, and people, and I tell my students, you know, we go to conferences because we keep discovering new stuff. You don't go to a conference with the same abstract. You know, you can't. I mean, we all know the rules. Anyone that's listening knows the rules of abstract submission, right? Never been published, never presented. And the fact that we do usually 2 abstracts every year to a conference, and we've been doing it since 2019, tells you how much new stuff we're getting. Well, there's 2 reasons. One, we just keep finding new stuff and our techniques are good. And this, you know, we're constantly, this is not just a semester that the students have a project and they get great at the end. This is an ongoing 12-month program. I mean, we just, like I said, we just met yesterday we had a Zoom meeting yesterday. Students got their Google Drives last night. They're working all this week, all summer. So this is a year-round process, but I just don't have time. You know, this last semester, you know, I'm bipartite, right? So I have 18 credits. I was teaching 18 credits and 3 lab sections. And it's not the problem of the university. It's difficult to get faculty up here. Our program's growing. And I have a hard time telling a student, no, you can't be in our program. So I tend to teach a lot. That doesn't leave a lot of room for writing. And I also wanted to make sure that we had enough data. Have to recognize that we have to wait every year for another group of data, because even though as a researcher, yeah, I would love to run lots of samples all at once, right? I'd love to be able to just do a bunch and just be in my lab and get a couple, you know, lab assistants and a couple students, and let's just crank out a lot of data. But that's not what we do here. What we're doing is we are generating a culture of research within the university program. And because we do that, as a researcher, I have to be patient for the next group of advanced students to give me my next piece of data, because it is not only important, and that's why we do abstracts, that's how we get the word out. I want people to know about this because we think it's really concerning, but it is also as important to me that as a researcher, but as a instructor, that I'm still giving the things that are so positive in my students, critical thinking, understanding, and I don't want to, we're just going to have to wait. And so because of that, we're at a point now that statistically I can maybe say something. So for anyone listening going, when are we going to see Grace's papers? We're really hoping to start submitting stuff to journals this coming, to One Health. We're going to try to submit something to One Health this coming fall semester. But it's not just lack of time, but also I do not want to put something out. And I tell my students that you want to put something out that stands. You know, you want to make certain that not only what you're saying is important, but that it's reproducible, it's valid, it's not just random error. You know, there is randomness. You know, when I first saw that ESBL, how did I not know that just wasn't random error? Because we have that. That's I always say that's why I can't find my car keys, because that's the random error that occurs in our life, right? And so because randomness is there, we don't want to make a huge firestorm over something that may just be random error. So so that's part of it too. So hopefully, now granted, we all know how the publication process is, but my goal is by this time next year, Louis, you'll say, and Grace just has their first periodical, their first article published with the students. But I will tell you that it doesn't take away from our students' excitement. I think sometimes knowing that it's just not, that it's a true program project. In other words, it's data been created by people that are now working in the lab somewhere, Providence Hospital, Kotzebue, whatever. But they all know that when that paper hits, that one of their 5 moose samples are there. One of the 300 of samples that we're going to talk about, they have their 5 is somewhere in there. And I think that's great. And I'm really proud of that.
That is great. And and you said that you'll be at Microbe this year in Atlanta. Do you know what day?
And I feel bad. I should know that. And what I'll do, Louis, I'll email it to you. So maybe you can, when you post this, you can say, because I would just got my confirmation for my hotel. And so I'm like, oh, when am I actually going to be speaking? And I want to say it's Thursday off the top of my head, but because it's in the middle somewhere. But I'll let you know, and I'll email it to you, and then you can then go, maybe when you post this, you can say that, this is when I'll be speaking. Yes. And I will also be speaking at the ASCLS conference, their JAM conference, but I'll be talking about One Health. And I'm actually speaking, and that is on their Thursday. They just changed me because I was on Tuesday of that day of that conference, but now it's on Thursday, and that's in July. That's July, I think 8th through the 14th or something like that. So I'll give you those dates. I'll make sure you have those I'll email them to you so that if you want to attach them, you can go ahead and do that.
Wonderful. And for the listeners, yes, when you see this, and stay tuned on social media. Once I have the information, I will go ahead and release it. Especially, and if you're attending Microbe and you want to stop by and meet Grace and listen, and definitely so you can have that information. Well, Grace, as we're coming to an end of the episode, I just wanted to ask you if there was anything else that you wanted to add for the audience.
Again, like I tell my students, your life should always be curious. I started this grad program late in my life because I was curious and I just wanted to know more, than the salesman, molecular and micro. And the only reason this program and all this research started is because I was curious. And so I think in our lives, as we move forward in our lives, if we can always maintain that curiosity, then our life will always have avenues that will open up to us. And I think sometimes when our life can be hard and, our economy isn't always great sometimes, people sometimes feel like you get stuck. If you can keep a piece of curiosity there, it may actually open a window, open a door, open a direction that you might not have never considered before. Remember that I was 55 and drove across Canada. And if I can do that, you guys can do stuff too. So just keep yourself open to curiosity. And I think in everything, micro especially, micro is always curious, but in every part of your life, I think you'll always grow.
Well said, and as I said before, I can listen to you talk for hours, and I love your passion, and I love what you're doing. So thank you so much for taking the time to be in Let's Talk Micro.
Yes, thank you so much. And maybe later on, we're going to be doing our goal because we just had our meeting yesterday. So we were talking about our protocol. So we're going to try to do 100 soil samples, just looking for 0157 in some of our areas of our parks that have a lot of berries, because we collect, we do that. We collect berries and that's also where our bear scat would more likely have been. And so maybe if I find something interesting, I'll let you know if you think you want to chat about that, because, that's, again, we're clinical microbiologists, but we forget that it all comes from the environment too. And so we have to cross those bridges. We're just not silo microbiologists. We have to think what's coming in our back door. So if you want to invite me on, me, I'll talk about anything. I'll talk micro every day. So thank you so much for inviting me. I really appreciate it.
My pleasure, and I will take you on that. All right, thank you. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about the research that Grace Leu-Burke is doing at the University of Alaska in Anchorage at their MLS program. I think it's definitely very unique, right? Very different from what other programs might be doing. And I really like it. The students are learning so much, getting so much knowledge, and really getting a grasp on antimicrobial resistance, which is such a dense topic. So, as always, right? Continue bringing that passion to what you do. It's so important. You do such great work. And stay tuned. Great things coming your way. And a little episode about AS Micro that I did, it's coming your way as well. So, thank you for everything. Thank you for listening. I am so grateful. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.